All right, welcome everyone to another Build a Cell seminar. Thank you, Anthony, for agreeing to give a talk today. I will let you take it away, introduce yourself, and share what you've been working on.
Well, thank you, Kate, for inviting me to the seminar series. And this is not my first presentation at the University of Minnesota. I'm now presenting, though, for the first time from Sweden. I don't know if you can see my horns there, but I wear that also in honor for Minnesota, which I gather the Vikings are currently undefeated there. And I am coming from Viking country, as we say, but you probably noticed from my accent, I originally come from Australia, the top of the world. Did my PhD at the University of Adelaide with Bob Simons. I later moved to Sid Altman's lab at Yale University and did an MD at Harvard and assistant professorship at Vanderbilt University before moving to Sweden at the bottom of the world. And I'm now talking to you right about where the bottom of this arrow is here, near the castle built by the first king of Sweden. So I picked an ambitious title for this talk, and we'll see how it goes. But I want to talk about the evolution of cells from the prebiotic RNA to the RNA world cells to peptide world cells and protein world minimal cells. And it'll be a bit of a personal journey through time. Following this timeline, I'll be talking about some of my work on the minimal size for catalysis by RNA, what caused the extinction of the RNA world cells. I'll talk about peptide world cells in terms of how were the amino acids originally selected by evolution, and what about the rejects that evolution didn't pick? Might they be useful to us? And I'll finish up with the protein world minimal cells with some investigations headed towards minimal components and ultimately building them, which is the goal of this group. So, of course, I want to emphasize that there's still much left to do, maybe more in this field than many other fields. The chemistry is quite problematic for synthesizing RNA prebiotically, both in terms of hydrolysis and stereochemistry. There are synthetic challenges in building RNA world cells using synthetic ribozymes that can polymerize nucleotides. Regarding the peptide world cells, it's still a big question as to how the complex ribosome evolved and translation. And in the protein world minimal cells, synthesizing self-replication from small molecules using defined macromolecules encoded by genes is still a huge challenge. So for the young people, plenty left to do here. Okay, prebiotic RNA. So how much do you need for catalysis? Of course, we're all very familiar with self-splicing RNA and RNase P discoveries of the catalytic role played by RNA by Check and Altman. Now, these RNA molecules. I don't know if you can see my pointer here. Is it visible, my pointer?
Yep, we can see it.
Good. Okay, so these RNA molecules are about 400 nucleotides in size. So it's a bit of a stretch for the prebiotic RNA world to imagine such RNAs evolving from prebiotic components. So during my PhD, I was at the right place and the right time when the Simons lab discovered self-cleavage of avocado sunblotch viroid, and I tested some related satellite RNAs for self-cleavage and proposed that a small conserved secondary structure and sequence present in some of these, but not all of them, might be responsible for the catalytic activity of the RNA. Now, this was only 52 nucleotides in size, and so it required some experimental verification, of course, but we were able to show that indeed this was sufficient for performing the transesterification reaction. So this provided the first example of a small RNA that could have catalytic properties and was getting closer to the sort of thing you might imagine appearing in prebiotic soup. And since then, I think Mike Yarris may hold the record. He's got like a 5 nucleotide ribozyme. So this is getting more plausible, I think. You're probably familiar with Shostak et al.'s review in Nature, where they talked about origin of life through perhaps an RNA replicase appearing. And they emphasized that for this to occur, you would actually need the simultaneous synthesis of 2 RNA replicases, because one wouldn't be able to unfold and replicate itself. And of course, subsequent brilliant work at engineering in vitro polymerization by ribozymes has had success in making polymerases, but they all tend to be bigger than about 150 nucleotides and still not terribly efficient. So evolving a key RNA replicase from RNA is maybe a big step for the first self-replicating organisms. So maybe we should be thinking about something a bit smaller. However, short oligos are impotent as catalysts, largely having a lot of trouble polymerizing, for example, guanines on poly C. Mike Yarris thinks there might have been some simpler systems like some sort of nucleotide cofactor catalyst. So I think it's still very much an experimental area to work on here. I am, of course, assuming that there was an RNA world because I think there is a preponderance of evidence for that. And I'm not going to go into that in this lecture, but happy to take questions on that. So what about these RNA world cells? These are presumed cells that are bounded by some sort of lipid bilayer containing RNA as their catalysts. Presumably these existed, as I mentioned, but they are totally extinct. And one has to ask the question, what caused their extinction? And a related question to that, of course, is why did the DNA and protein molecules evolve to replace the RNA world with the DNA, RNA, protein world? I'm happy to take questions at any time, by the way, as we go. So there's not much disagreement that DNA is much more stable than RNA, which is advantageous for genes. So we can readily understand why DNA was selected by evolution. It even looks very similar to RNA chemically. You have replacement of this 2' hydroxyl with a hydrogen, and this prevents the intramolecular attack on the phosphate, which is quite favored, making RNA quite unstable, particularly at high pH, which this doesn't happen in DNA. Okay, but what about proteins? So here, most people would say the reason we have proteins for our catalysts is that the 20 amino acid side chains are better for catalysis than RNAs for side chains, the nuclear bases. That seems reasonable on first examination. There are no pKa's here near neutral pH. And you've got a lot of different side chains on the common 20 amino acids, but they're far from perfect, actually. In fact, they even lack an electrophile. There is no electrophile amino acid side chain. So nature has to rely on metals pretty much for that function. And certainly RNAs can use metals. So could there be another explanation here? So I want to bring up RNA modifications, which often don't get much credit in biochemistry. So there are a lot of them, an awful lot of them. And if you look at the overlap in RNA modifications between the eukaryotes, the bacteria, and the archaebacteria, there are still quite a few. So these were presumably present in LUCA, the last universal common ancestor. And if we look amongst these conserved ones, well, one of them has an interesting side chain anyway. It has a carboxylic acid. So it's possible if you go back even into the RNA world, you might have had this one with the carboxylic acid and possibly had others. You could have had, for example, amino groups and other nucleophiles. So I got thinking about this, looking at the statue in our courtyard here at the Biomedical Center, which shows a sort of more complex arrangement of the side chains as it goes up, maybe sort of indicating evolution. And I thought, well, why couldn't riboorganisms evolve modified RNAs that had side chains that could do everything the amino acid side chains do? Why would it be necessary to evolve protein? Any path to protein in evolution is very complex. In fact, looking through the literature, there's no real agreement on what it was. There must have been so many steps, but many of them are similar to the steps I've put together here, where I show in red pure RNA molecules performing the reactions and then sort of moving to more and more containing amino acids and peptides as we get down to the ribosome. But it's obvious that there are many steps involved to evolve the complex translation machinery, including the ribosomes, the mRNAs, tRNAs, factors. to get to a working translation system, not to mention the genetic code. So this seems like a rather complicated and long-winded and slow route to getting the side chains you want for catalytic reactions. So what problems might RNA have encountered if it was the first catalytic molecule and it wanted to evolve suitable side chains for biochemical reactions? Well, one obvious challenge is that because RNA is a highly charged negative polymer, it would have extreme difficulty creating membrane pores, transporters, and scaffolds for allowing small molecules to be taken up from the environment and to remove waste from the cell. Mike Yaros published an interesting example of an RNA that can interact with a lipid bilayer to somehow make it more permeable. But I think you can see my point that this is quite a challenge for an RNA molecule. Secondly, hydrophobic cores are necessary for some enzymes. And it's very difficult for RNA molecules to completely exclude water from their active site because of their charges. And I highlight a particularly important reaction, which is the ribonucleotide reductase radical mechanism, which is needed for DNA synthesis by life. So the way life makes DNA, it starts with RNA, which is actually kind of a difficult way to do this. And it converts it into the DNA via a free radical mechanism. And these free radicals would immediately react with any water around. So the enzyme has to keep a very hydrophobic core. And this has actually been cited as a potentially a problem for ribozymes. Nobody has evolved in the test tube a ribozyme that can do this. And an argument even that maybe, although DNA looks so similar to RNA, maybe protein came first before DNA as a way of being able to catalyze this reaction. So clearly the charge on RNA is a problem for two reasons. But couldn't the charge on RNA just simply be removed? I mean, it seems a reasonable idea. So the idea would be if you would perhaps alkylate this phosphodiester group in the RNA. And in fact, based on the chemistry of RNA, phosphates are more susceptible to alkylation than most other RNA groups. So it's not a terribly implausible reaction. Whoop. So let's see, what have I done here? Okay, but it turns out if you look at nature, even in today's DNA, RNA, protein world, this phosphodiester is never modified in RNA. So this is all RNA modifications that have been found. The RNA phosphodiester is frozen in evolution. It is never modified. There's actually one phosphodiester modification which occurs in DNA, which is to a thiophosphate, but DNA is a different story. I'll get to that. So that got me thinking, this is something difficult chemically here. And so I spoke to my chemist collaborator, Marek Kwiatkowski, and asked him about this. And he said, oh yes, of course, he said, because he knew that if you modify this negative charge into a phosphodiester in RNA, this um, phosphodiester also then becomes susceptible to intra attack from the adjacent hydroxyl group. It turns out that this negative charge, which is delocalized on this group, is critical for repelling these electrons from the adjacent 2' hydroxyl group. So this reaction takes at neutral pH around about an hour or so to happen. So it turns out that phosphotriester RNA is unstable, so it really was impossible to remove this negative charge from RNA and wind up with a stable molecule. DNA is different, however. The DNA phosphotriester is more stable. Hence, there had to be another way to evolve biopolymers with greater hydrophobicity, a more convoluted way, which is going through this very long-winded evolution of translation. So that's my theory there. Moving along to peptide world cells. How were amino acids selected, and might the rejects be useful? So directed evolution of binders in vitro is a very powerful technique invented by Joyce, Gold, Shostak, and for peptides, Dower. And the way this works is reiterated steps of mutation, selection, and amplification to have Darwinian evolution in the test tube. And sometimes the initial library is so diverse you don't even need to introduce a mutation step. However, if you want to use this process to evolve drugs, this can be a challenge. So, as you know, RNases are susceptible to nucleases and they're cell impermeable. Peptides are susceptible to proteases and they cause immune responses. And although the body does this process extremely well with antibodies, the antibodies are not ideal drugs to give. They have to be injected and they're very expensive. So my idea was, what if we could take advantage of this beautiful directed evolution technology, but instead of evolve high affinity binders and drug-like molecules made of nucleotides or peptides, what about unnatural amino acids? Ones that would allow rewriting the genetic code we have in Swedish here. Instead of DNA makes RNA makes protein, we want DNA makes RNA makes peptidomimetic. So I joined Steve Blacklow's lab at about this time with this idea. And we decided the best way to go about this was to develop a purified histag translation system. In this way, we could avoid the challenges posed by competition with the natural amino acids used by pretty much everybody else in the translation systems at the time. They were using crude translation systems, both E. coli and in eukaryotes like the rabbit retic. So I visited Herb Weisbach's lab for a couple of weeks to learn about this, he was the first to purify a complete translation system that makes full-length proteins. And we put together a his-tagged translation system using E. coli initiation and elongation factors because it's a lot simpler than the eukaryotic translation system. And importantly, the day before we published this, we filed a provisional patent application. We also incorporated some unnatural amino acids in this. And this was helped through a collaboration with Virginia Cornish at Columbia, who was an expert in synthesizing PDCA amino acids via the Schultz-Hecht approach for ligation onto tRNAs lacking the terminal CA using T4 RNA ligase. And so you would then generate an aminoacyl tRNA whose protecting group could be removed photochemically that would allow installing essentially any R group you want in the amino acid. So we wanted to test translation with N-alkyl amino acids particularly. And the reason I wanted to do this was inspiration from cyclosporin A, which you probably know is one of the most widely used immunosuppressive drugs. It is a cyclic 11 amino acid peptide drug that is importantly orally available. And this contains 7 N-methyl amino acids. The methyl group reduces its protease susceptibility, makes it more hydrophobic. So we wanted to see if what would be the effect of these N-methyl groups on translation, and we picked the only N-alkyl amino acid in the genetic code, which is proline for comparison, and also alanine and a bigger chain on the amino group, N-butyl amino acid, and a few of these others, and put them in at 3 different codons on 3 different RNAs to control for those potential effects. So you can see the results in this slide, looking at incorporation in the 3rd position in this tetrapeptide. So if you look at the amino acids on the left, alanine, phenylalanine, these gave quite good yields of tetrapeptide. When we moved to proline, sometimes the yield dropped a little bit, but not much. Then look what happens when we go to N-methyl amino acids. There was a big drop here. Although it worked a little bit better here on tRNA asparagine and tRNA alanine, suggesting that some matching of the tRNA and amino acid might be helpful. And shockingly, the N-butyl amino acid was not incorporated at all, even although this is a purified translation system, essentially waiting as long as you like to incorporate it. So that was surprising. So our conclusion was that incorporation is dictated by amino acid sterics. And to some people in the non-ribosomal peptide synthesis field and also synthetic chemists, this is not a huge surprise. So in the non-ribosomal peptide synthesis, generally you don't go much beyond an N-methyl group in your peptides. You don't see these types of groups. And also the synthetic chemists told me that when they do solid phase peptide synthesis, they know that proline incorporates better than an N-methyl amino acid, which they explain that it's kind of a privileged structure where the lone pair on the nitrogen is sort of sticking out in the proline here, which makes it a bit more reactive than in the case of the freely rotating methyl group on the N-methyl amino acids. So this brings us to another origin of life puzzler. Why is proline the only N-alkyl amino acid of the 20 amino acids in proteins? Well, it's apparently not due to low abundance. N-methyl amino acids are quite abundant in the famous Miller-Urey spark experiment, where chemicals were sort of heated up in a prebiotic reducing atmosphere in a flask. And at the end of the day, amino acids were detected, and there were plenty of N-methyl amino acids as well as normal amino acids. Also, there's a famous meteorite that hit Murchison, Australia when I was 7 years old. Murchison is about 100 miles north of where I was living in Melbourne. So this was actually a near miss astronomically. And this meteorite is quite famous because it contains a lot of carbon. And they analyzed this meteorite and found that it too contained a lot of N-methyl amino acids, about 10 of them, as well as about 10 of the proteinogenic amino acids. So are you with me? Why is proline the only N-alkyl amino acid in proteins encoded by your genomes today? Anybody? Anybody there? I can't see your screen right now. Why is protein the only N-alkyl amino acid in your genetic code? Based on what I've told you.
I don't think anyone knows. I'm looking at the chat and I'm seeing confused faces.
Okay, well, my conclusion is because it's more reactive. It's that
Hmm.
simple. Proline is more reactive than N-methyl amino acids, and nature picks things that work. So that's my conclusion, and the wonderful thing about proposing ideas about the origin of life is nobody can ever prove you wrong. Okay, but let's move on to some other unnatural genetic codes. So we made a rudimentary genetic code incorporating 3 side chains that are not present in nature: an alkyne, a methylserine, an alkene. And we put these 3 in a row into a tetrapeptide and confirmed it by co-migration with synthetic standard. What was a little bit disturbing about this first de novo genetic code was that there was only 80% average coupling per unnatural coupling, despite no competitors in this purified translation system. So there was some sort of problem with the system that we hadn't anticipated. So this led me to a collaboration with Måns Ehrenberg's lab in Uppsala, where they had a in vitro kinetics system of incorporating. They could put Phi tRNA Phi together at 54 amino acids per second, which is even faster than the in vivo rate. So I wanted to check out some of these unnatural amino acids in this system. So if you just put a single N-methyl group on the phenylalanine tRNA, it's loaded from 41 seconds, 41 couplings per second to 0.005 per second, 8,000 times slower. So they said, this is remarkable. This is practically infinity. And I said, why did I have to travel from America to Sweden to do this experiment? I could have just hand pipetted it. It was that slow. It's taking more than a minute to get in here. So then we wondered, well, how does proline get in? And we put proline on the tRNA Phe, and even then it was a bit slow, 2 per second. So we thought it might have to be matched with its tRNA, as Oki Uhlenbeck had shown, a matching of the tRNAs and the amino acids. And indeed, if you put it on its pro-tRNA, it goes in at 9 per second, but still slower than Phe. So this told us a couple of things about basic translation, which is that incorporation of different amino acyl tRNAs is not the same rate. And it also told us that the rate limiting step in protein synthesis was not accommodation. So at the time, it was thought that the movement of the tRNA after it was released from EFTU so that it accommodates close to the peptidyl tRNA was rate limiting. And you might think that because it's like a big movement. But in fact, our data argued that it was this peptide bond formation step that was rate limiting. And that comes to the question as to why weren't our reactions 100%? Even although there are no competitors. So if you imagine that this reaction is slow and can even take a minute for some of the worst cases, like N-methyl amino acids, that actually gives you time for the peptidyl tRNA to actually drop off the ribosome, and then you're done if that happens. So we think the competing reaction here is drop-off, and we had to go through a lot of kinetic studies and optimizations in Uppsala to find the best matches of amino acids and tRNAs to reduce this. And it turns out you can reduce it quite effectively if you do that. Hiro Suga has also been very prominent in this work. So I wanted to mention utility, which I had in my title. Excuse me, I've got a bit of a cold.
Bless you.
Thank you. So this provisional patent that I wrote the day before we published our purified translation system, I eventually got a patent on that, and Ra Pharmaceuticals was interested in that patent and used it, licensed it together with some other technology to found a company for peptidomimetic evolution, founded in 2008 in Boston. Jack Shostak and co-workers cleverly demonstrated that they could perform this directed evolution of peptidomimetic libraries to obtain a useful peptidomimetic inhibitor at the same time as Hiro Suga also did some directed evolution experiments, and so that established proof of principle. The company went public in 2016 when it had 40 employees. And one of the great things they did was they designed a peptidomimetic inhibitor of complement C5, which bound to the other side of complement than an existing inhibitor. And this showed in up to phase 3 clinical trials efficacy for treatment of the neuromuscular disease myasthenia gravis, and is hopefully going to be useful for other autoimmune diseases. So this led to a US $2.1 billion, that's billion, not million, merger agreement with Belgium's UCB, and Hirosuga's company Peptidream also was sold for even more, I believe. So these are now at the forefront of synthetic biology in actual drug discovery rather than just synthesizing drugs. So these unnatural amino acids, though rejected by nature, have great utility to us precisely because nature now doesn't know what to do with them. Okay, let's see here. Okay, so there were some other applications of my ribozyme work, gene shears by Hasselhoff and Gerlach, and my external guide sequence RNAs in the Altman lab used to target RNAs for cleavage, but these haven't panned out therapeutically yet. So I want to get to the last part of my talk now. I see I'm about 45 minutes in, so I'll keep this kind of short here. So what about the minimal components for the protein world, such as LUCA? So this was sort of inspired by the Tomita Hutchinson and Venter E-cell paper, which has now been cited more than 1,000 times, where they put together a list of what they thought would be essential minimal components from mycoplasma, although there are a few omissions here, such as DNA polymerase, some tRNAs, and some translation factors. So George Church and I thought we would put together a more complete list based upon known biochemistry towards synthesis of a minimal cell to get people starting to think seriously about this, and maybe sort of raise the question, how close are we to doing this? And of course, there are in vitro synthesis systems, purified systems for DNA synthesis, for RNA synthesis, transcription, or for protein synthesis. Some of them have been combined together, transcription and translation. So the idea was if you could somehow package these in membranes and get transporters right to get small molecules in there, maybe you could fire this up to go a little way. So we came up with 151 genes required, and interestingly, 96% of them were for translation. The ones shown in yellow were unknown in 2006, and interestingly, all were for RNA modification enzymes, some of which had not even been identified for the ribosomal RNA modification. So it shows you how this field of biochemistry was behind the rest. But since then, all but one of the RNA modification enzymes have now been found. So we focused on these, particularly ones that modify around the peptidyl transferase center loop in the 23S ribosomal RNA of E. coli. And these were identified as critical by mix and match experiments by Green and Noller in vitro using unmodified RNAs mixed and matched with sort of broken up versions of the wild type RNA with the modifications. So one thing is, if you want to build a well-characterized ribosome like the E. coli ribosome, maybe you need to introduce several of these modifications at once at the peptidyl transferase center. And we succeeded in doing that using a naked 23S ribosomal RNA, which is actually a reasonable feat because you worry about whether the RNA alone would fold correctly in the absence of the ribosomal proteins. And also some of the modifications are thought to occur late in the assembly process. But this we could get working very efficiently with 4 of those 6 critical region modifications based on the reverse transcriptase assays shown here. Okay, so then we wanted to know why they were critical. And here we had a collaboration with Janis Remi in Estonia, where we KO'd all of these blue ones, except for the dihydrouridine one, which still hasn't been identified, but is not thought to be important, and this partially modified HO5C here. And remarkably, we could make cells that were viable with simultaneous KOs in all 5. And they grew quite well, less well at 20 degrees. So this was a surprisingly minimal phenotype for these critical, so-called critical modifications. And we thought this may be because Green and Noller's assay was very different. An in vitro assay, and which involves folding, etc. So we wanted to know what these critical modifications actually do. So we did some biochemical assays, some sucrose gradients, and we found that the 70S assembly is quite poor if you lower the magnesium to 5 millimolar. It works quite well at 10, but 5 was more physiological on it, and there's a big problem. Interestingly, dipeptide synthesis was unaffected in a number of assays we tried, but we could see a small but significant effect in tripeptide synthesis. And when we did this in vivo, synthesizing beta-galactosidase, we showed that elongation, this blue bar here, was significantly slowed, consistent with a defect in translocation. So we think these critical modifications affect the last step of ribosome assembly, which is known that PTC maturation is the last step and important for assembly. And we think, interestingly, it has an effect on translocation, which maybe involves more large-scale movements of the ribosome than dipeptide synthesis. So that was a bit unexpected also. And finally, Kate's grant and the seminar series talks about pure that makes pure. So we started working on our purified translation system sort of independent of the UITA lab and published at the same time our simplified histag translation system. And then when we saw theirs, of course, we asked for all of their synthetases and started working on this, and that was very exciting. So we thought how to put together a genome for a theoretical minimal cell, where to start. So we thought start with Weisbach's purified translation system, the one that UITA histagged, which is 30 of the 151 genes, but actually half of the minimal genome in size. And we showed that you could make from 3 pieces of this prepared by BioBrick cloning large plasmids for production of whole bunches of these translation factors in vivo, and we're able to reconstitute protein synthesis in vitro from the purified histag proteins overexpressed in these 3 strains. We then put them together into a bacterial artificial chromosome, and I show you a plug here for our next synthetic biology manual where we put it on the front cover here, the 65 kb construct of 30 cistrons, and showed that it could express all of the proteins both in a crude translation system and in a nice collaboration with Christophe Danelon in a pure translation system using pure FREX. But what they discovered was that there was a per codon processivity loss that is about 50 to or 5 to 50 times greater than seen in vivo, and less processivity than we would need for break-even synthesis in vitro. It wasn't really proportional to the length of the proteins made, and they weren't able to identify what the problem was in the translation system, although they analyzed a lot of different possible causes. So I'll leave you here then with my final slide, that there's much left to do in all aspects that I've talked about today, but certainly it's a very interesting journey. Thanks very much.
So awesome to hear your talk, Anthony. That was amazing. We already have plenty of questions in the chat if you're able to access that and read them out loud, or I could help do that. And I'm sure there's more coming. So um, let's see. I'll read out the first one. This is from early on in your talk. Why 20 amino acids? The codon table, even with wobble, has room for at least twice as many. When RNA world transitioned to proteins, why didn't nature utilize more side chains?
Yeah, well, I guess at the early stage of building the code, there would have been even less, of course. So why did it move up to 20 might be another way of phrasing the question. But also, when you look at the degeneracy in the genetic code, sometimes you can reduce errors by having a little bit of degeneracy in there. And maybe if you have to get, up to, if you have perfect wobble pairing and a different amino acid for every codon, then accuracy could be a problem. That's just speculation. If that answers the question.
If it doesn't, I'll expect Carl Engelhardt to voice up. All right, let's see. So Ava says, why didn't life finish the transition from RNA world to protein, the PTC, ribozymes, etc.? Why do those molecular fossils still exist versus replacing those functionalities with more chemically versatile and stable proteins?
Well, life has been pretty darn effective at replacing RNAs. Even recently, there have been examples found in the RNase P field where even some of the RNase Ps have been completely replaced with protein. So protein seems to do a better job, but it might be hard to replace certain RNAs with protein. And we thought about that particularly with the ribosome. The large 23S RNA and 16S RNA in the ribosome probably perform most of the catalytic functions of the ribosome. And if you start interfering with that, you might wreck the ribosome, if that answers your question.
I think it does. Let's see. So Hannah Bergenhold says, we have DNA-based pores like, hang on. Oh, like the work from Simul Lab. They're all artificial, of course, but wouldn't it be possible for nature to invent nucleic acid membrane pores too? Could you speculate why didn't that happen?
Well, the negative charge is a problem. Mike Yarris, as I said, made them leaky. I don't know about these DNA pores. Perhaps you can send me a reference in the chat here, or it could be an area for experimentation. But I think integrating nucleic acids in defined ways into membranes is pretty tricky.
Karen Verge says, awesome talk. I think we all agree on that. When you think about steric hindrance limiting translation of interesting but bulky residues, did you ever think about doing directed evolution on the exit tunnel? Isn't that where most of the steric caused stalling happens? Steric caused stalling. There we go.
Well, I think by the time you get to the exit tunnel, you're almost done. So you've probably got through the tough parts. I think just forming the first peptide bond can be very tricky. So we've used biotinylated amino acids, for example, and they don't work very well. They don't fit on EFTU very well for delivery to the ribosome. So I think the tougher steps might be earlier on in getting into the chain. You can extrude biotin though out of the tunnel, and that's how we did some of our first selection experiments.
And we have a clarifying question from Gabriel Garcia, who again says, fascinating talk. And again, we all are on board with that. Sorry if this is a naive question. All questions are perfect questions, Gabriel. I don't know the literature well enough. What is the difference between your purified translation system and the Japanese pure?
Yes, so the original purified translation system we made just had the 3 initiation factors and the 3 elongation factors. It didn't have the amino acyl tRNA synthetases or the release factors. One of the reasons was I only wanted to make short peptides. A second reason was I was a little bit scared about proofreading activities of the synthetases, which hadn't been so well characterized. And it's known that some amino acyl tRNA synthetases can proofread off incorrect amino acids from the tRNAs.
Okay. Well, let's see. We're cruising pretty fast here, but let's see. Um, how did you assemble the 50S ribosome subunit? Were they in lysate pure or the two-step reconstitution? Um, what have you tried? It's from
Yeah,
Mohammed
we,
Sofi.
we use the classical two-step reconstitution where you use different magnesium concentrations and temperatures in a purified system.
And then again from Mohammed. Oh,
oh, sorry, I'm not sure what experiment you're talking about because I don't know that I even presented that experiment actually.
mm. Mohammed, if you're still around, I know we're pushing our 12 o'clock
Yeah.
limit.
I wanted to, so it's the one where you added the RNA modification enzymes to the 23S rRNA. Yeah. How did you make those ribosomes?
Yes, then there we didn't reconstitute the ribosome.
Did you
We
just
used
only do the RNA?
yeah, we used naked
I
RNA,
see.
which
Okay.
it can fold fairly well.
Into its native structure?
Close, yes.
Oh, that's cool.
We have another one from Mohammed here, more generally. Can you speak on ribosomes making ribosomes? How would you go about it?
Well, George Church has done more work on that, making the ribosomal proteins from genes he made by PCR. This is an area we're working on at the moment, actually. So if you look in the literature, what is actually required for a ribosome? Which of the proteins in particular? It's not very clear. And do you need all of them? Um, Most of the proteins also are quite short, which is an advantage.
Is the structure of the 23S rRNA by itself functional? You said it folded into the right shape.
Uh, There were some original publications saying it was, and then they were rebuked by Rachel Green and others. So, to my knowledge, the RNA alone is not functional in catalysis.
Okay. All right, we are past our time, so I want to give a last call for questions. I know that we could talk about this all day, and I'm sure some of you would love to. In the chat, we're seeing a lot of thank yous and great talk, and I wholeheartedly agree. That was awesome, Anthony. Thank you so much for sharing your work with us. And thank you everyone else for attending our Build a Cell seminar this morning.
Well, thanks so much, Carlisi, and everybody else.
All right. Have a great day.